Hello students, this is Mrs. Sonal Patil. In the previous class, we have completed up till Hershey-Chase experiment. Today, we will study about the properties of genetic material. Now, what must be the properties of the genetic material? Here, we will study DNA versus RNA. It should be able to generate its replica. So it should produce its own replica. That is the first property of the genetic material. Secondly, it should be chemically and structurally stable. So chemically and structurally, the genetic materials should be stable. Thirdly, it should provide the scope for slow changes that are required for evolution. Nextly, it should be able to express itself in the form of Mendelian character. Protein does not fulfill the criteria, hence it is not the genetic material. RNA and DNA fulfill the criteria. So these are the properties of the genetic material. So the genetic material should be able to produce its own replica. It should be chemically and structurally stable, should provide scope for slow changes which are required for evolution, and it should express itself in the form of Mendelian character. So these are the properties of a genetic material. Now let us see RNA. About RNA we'll study how RNA is unstable. 2' OH group present at every nucleotide in RNA is a reactive group and makes RNA liable and easily degradable. So 2' OH group, 2' group, which is present on every nucleotide in the RNA is very reactive and it is making RNA liable and easily degradable. RNA also acts as a catalyst, hence it is reactive. RNA is unstable and mutates much faster. Consequently, the viruses which are having shorter lifespan mutate and evolve faster. But RNA is the first genetic material in the course of evolution. RNA is found to be the first genetic material in the course of evolution. So hence RNA is unstable, whereas DNA is more stable. How it is more stable? Let us see. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle. In the Griffith's transforming principle, we have seen about the stability of the genetic material. So stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of the genetic material. We have seen this from the Griffith's experiment. Two strands being complementary, if separated by heating, come together when appropriate conditions are provided. So by heating, the two strands of the genetic material get separated, but when we provide appropriate condition, they again come together. Presence of thymine in the place of uracil confers additional stability to DNA. So thymine is present instead of uracil in DNA. So it is providing additional stability to DNA. DNA is chemically less reactive and structurally more stable when compared to RNA, and hence this tells us that DNA is more stable than compared to RNA. Now let us see which is the better genetic material, DNA or RNA. Now presence of thymine at the place of uracil confers more stability to DNA. We have seen that both DNA and RNA are able to mutate. Both can undergo mutation. In fact, RNA being unstable mutate at a faster rate. So RNA being unstable, it is mutating at a much faster rate. RNA can directly code for synthesis of protein, hence easily express. It can easily express because it is directly coding for the synthesis of protein. DNA, however, depends on RNA for protein synthesis. We have seen in the central dogma. Okay, so DNA is dependent on RNA for protein synthesis. The protein synthesis machinery has evolved around RNA. Both RNA and DNA can function as genetic material, but DNA, being more stable, is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. Hence, from the above points, we come to a conclusion that DNA is a better genetic material than RNA. Important question from exam point of view. So that is about DNA and RNA, which is a better genetic material. Next, coming to the DNA replication, a very important question from exam point of view. Replication of DNA will be asked. The parental DNA molecule produces two identical copies of daughter DNA molecule is called DNA replication. DNA replication means what? It is producing its own replica. Its own copies are being produced, so it is called as DNA replication. Now, when does this DNA replication take place? We have already studied in the first PUC during when we have studied about the cell division, mitosis and meiosis. Okay, before that we studied about the phases which are occurring, G1 phase, S phase and G2 phase. So this DNA replication is occurring during the S phase. So it takes place in the nucleus of a cell before cell division, that is before actual cell division occurs. In the S phase of interphase stage, DNA replication is occurring. And how this process will start? The process of replication begins at a specific point on the DNA strand. So there are specific points present on the DNA strand which are called as ori site or site of origin, or it is called as initiation site. So these are the site where the process of replication begins. The double helix starts unwinding from the origin, catalyzed by unwinding proteins and helicases enzymes. So remember, process of replication will begin at ori site, and from there, from the ori site only, the unwinding of the DNA double helix starts, and it is catalyzed by helicases enzyme and unwinding proteins. Now next step, what happens is that the hydrogen bonds which are present between the two strands, you know there are hydrogen bonds present between the two strands of the DNA, these starts dissolving. They are dissolved by DNA polymerase 1 and topoisomerases. Okay, so hydrogen bonds starts dissolving. Then when hydrogen bonds dissolve, the double stranded DNA is now getting separated to form two single strands. Now, since earlier it was double stranded, hydrogen bonds are dissolved. Now we will get two single strands of DNA. This mechanism is called unzipping of DNA. Now after unzipping, the double helix is separated, it gets into Y shape. It gets a Y-shaped appearance. It is called as replication fork. Y-shaped appearance is got to the DNA and is called as replication fork. Both the separated single-stranded DNA acts as a template. So each one is acting as a template on which new strands are going to be synthesized. So two strands are separated, each one is acting as a template, okay, and on this new strands will again be synthesized. Now next what is happening is that before addition of the DNA nucleotides onto the template, a short segment of RNA strand is synthesized called RNA primer. RNA primer should be synthesized before the addition of the DNA nucleotides. So before the addition of the DNA nucleotides onto the template, a short segment of DNA strand is synthesized, which is called as RNA primer. It is catalyzed by RNA primase enzyme. Here base pairing would be possible because adenine to uracil. It is possible between adenine to uracil. The length of RNA primer is maximum of 10 nucleotides. So RNA primer is nothing but it is a short sequence of 10 nucleotides which is added here, okay, before the addition of the DNA nucleotides, and the pairing of the base pairing starts will be possible between adenine to uracil. Further, addition of DNA nucleotides takes place to the exposed bases on the template continuously towards the replication fork. So on one strand it is going towards the replication fork and they are added continuously from the end of the RNA primer. It is called as leading strand. So on the leading strand the bases are continuously added. Okay, it is catalyzed by DNA polymerase 3. On the other hand, DNA nucleotides are added discontinuously away from the replication fork. So from the now in the leading strand towards the replication fork, the nucleotides are being added and they are added continuously. But on the lagging strand, the DNA nucleotides are added discontinuously and they are away from the replication fork, and that strand is called as a lagging strand. These fragments of DNA are called Okazaki fragments. So discontinuously the nucleotides are added and they are in the form of fragments, because of which they are called as Okazaki fragments. Even the Okazaki fragments are also formed only after the RNA primer. So Okazaki, since the scientist has been discovered this particular fragment, so the name is given by him, Okazaki fragments. Later RNA primer is replaced by DNA nucleotides and the Okazaki fragments are joined by DNA ligase enzyme. You know, Okazaki fragments were in the fragments, and these fragments should be joined, and that is done by DNA ligase enzyme. The synthesis of DNA proceeds towards both the directions. It is proceeding towards both the directions from the origin, hence it is called bidirectional. In both the daughter DNA molecules, one strand is parental and another is newly synthesized. So one is, we have seen in the first step there, DNA unwinding starts, it is separated into two, that is nothing but double stranded DNA separates to form two single strands. So here each one is acting as a parental strand on which newly the one more strand is being synthesized. Hence DNA replication is called semi-conservative method proposed by Michelson and Stahl. So DNA replication is called as semi-conservative because here one strand is parental and one strand is newly synthesized. So semi-conservative it is called as. So here in the diagram it has been shown the replication or the DNA replication. You can see that here it has been shown. So this is a Y-shaped fork which has been formed. So remember the steps of DNA replication. The process of replication begins at a specific point, okay, which is called as Ori's site, and then what is happening is that the double helix starts unwinding from the origin and it is catalyzed by helicases enzyme. Helicases enzyme is catalyzing the unwinding. So unwinding of the DNA starts. Fine. Then the double stranded DNA is now getting separated to form two single stranded. Two single strands are being formed of the DNA. This mechanism is called as unzipping of the DNA. As a double stranded or as a double helix is separated, it gets Y shape. Can you see here? Double helix has got separated and it is giving a Y shaped fork which is called as replication fork. Both separated single strand, you can see over here, acts as a template on which newly synthesized strands are they are going to be synthesized. Okay, then primer is been added before a short strand of nucleotides of 10 nucleotides is added. Here RNA primer is been added by using the enzyme called as RNA primase. Okay, here base pairing would be possible between adenine to uracil. Length of the RNA primer is maximum of 10 nucleotides. Further addition of DNA nucleotide takes place. It takes place to exposed bases on the template continuously. So you can see here the nucleotides are added continuously here. So it is called as leading strand. Whereas on the other strand you can see that it is in the fragments. The nucleotides are added in the form of fragments and these fragments are called as Okazaki fragments, and later these fragments are joined by DNA ligase. Ligases enzymes are involved over here for joining of the Okazaki fragments. So this is about the DNA replication. Again, very important question. For 5 marks it will be asked. So please remember this DNA replication along with the diagram and remember the steps of the DNA replication. For today we will stop here, and in the next class we will study about the experimental proof for semi-conservative nature of DNA replication. Again a very important question. For today we will stop here. Thank you.